Hello everyone and welcome to the new session. Myself, Dr. Narkati working as Assistant Professor, Department of Genetics and Plant Breeding, School of Agriculture, SR University, Bangalore. So, today's session is about tomato breeding. So first, we will see what are the benefits tomato. So this tomato will improve the vision. Tomato will prevent the gallstones. It lowers the hypertension. This tomato is good for digestive health. It helps to manage the diabetes. It prevents urinary tract infections. The tomato counters the bad effect of smoking. It reduces cholesterol levels and protects the heart. So these are the general benefits of eating tomato. Now we'll move towards our main topic, that is tomato breeding. Among the vegetables, maximum attempts have been made to improve this crop because of its short duration, easy cultivation, and large number of seeds per fruit. So this has made it an ideal crop for many research workers. An exclusive tomato genetics cooperative located in the University of California, USA, has published genetic information on the crop and maintains a large number of collections which have already been thoroughly catalogued. Tomato is originated in Peru, Ecuador, and Bolivia on the basis of availability of numerous wild and cultivated relatives of tomato. From its center of origin, the tomato first moved to Mexico for domestication and cultivation. What are the uses of tomato? So, tomato is also rich in medicinal value. The pulp and juice are digestible, mild aperient, a promoter of gastric secretion and blood purified. It is reported to have antiseptic properties against intestinal infestations. In the present days, it is gaining more medicinal importance because of antioxidant property of ascorbic acid and lycopene content. It is also an important source of beta-carotene and valued for their color and flavor. Thus, today it is one of the important raw material for multi-million food industries. Tomatoes are also called as poor man's apple. Tomato is consumed either fresh, cooked, or processed into various products like juice, ketchup, sauce, whole canned fruit, and is the main constituent of pizza. Now, taxonomy. So, family Solanaceae. As we know, the tomato is Solanaceous crop. Linnaeus called it as Solanum lycopersicum. Miller called it as Lycopersicum esculentum. Now restored to Solanum lycopersicum. This lycopersicum in that first Eulycopersicum, the red-fruited edible species with carotenoid pigmentation, annual and self-compatible. For example, Lycopersicum esculentum and Lycopersicum pimpinellifolia. Second, Eriopersicum, that is green-fruited species with anthocyanin pigmentation and self-incompatible. That is Lycopersicum hirsutum, Lycopersicum peruvianum, Lycopersicum chismeni, and Lycopersicum chilens. Like scientific classification of tomato. So kingdom Plantae, then it belongs to Angiosperm eudicots, order Solanales, family Solanaceae, genus it is Solanum, and species is Lycopersicum, botanical or binomial name Solanum lycopersicum. Synonyms Lycopersicum esculenta. This Lycopersicum cerasiforme is the immediate progenitor and Lycopersicum hirsutum is the ultimate progenitor of the cultivated tomato. Interspecific cross between Lycopersicum esculentum and Lycopersicum pimpinellifolium are easily made. Embryo abortion may occur between Lycopersicum esculentum crossed with Lycopersicum peruvianum. However, this can be overcome through embryo rescue technique. Chromosome number. It is true diploid, that is twice n is equal to 24. All species of Lycopersicum have twice n is equal to 24. Some other species are there like Lycopersicum pimpinellifolium. It is fusarium wilt and early blight resistant. Lycopersicum peruvianum, it is resistant to leaf curl virus. Lycopersicum cheesemani, it is resistant to salt. Lycopersicum hirsutum, it is fruit borer resistant. And Lycopersicum pennelli, it is resistant to drought. Okay. Now, importance of tomato. As this tomato is one of the important crop, it is the second most consumed vegetable after potato in the world. First, in terms of total contribution of nutrients to human diet among the vegetables. It consumed in fresh form and various processed forms like soup, sauce, ketchup, paste, and like canned fruit. Tops the list of processed vegetables. It is ideal model plant for physiological, biochemical, cytological, and molecular studies. It has a very few competitors in value addition chain of processing. Rich in citric and malic acid. It contains minerals like Na, P, K, calcium and magnesium. So tomato is high in mineral content also. Rich in mineral content also. So as far as botany of tomato plant, so this tomato plant is characterized by two types like determinate growth habit and indeterminate growth habit. So determinate type, inflorescence occurs more frequently in almost every internode until the terminal ones are formed and elongation ceases at this point. In other words, it may be defined as self-topping and the main axis terminates with a flower cluster. And indeterminate, the inflorescence cluster occurs at every third internode and the main axis continues growing indefinitely. What are the objectives of tomato breeding? Breeding for earliness, breeding for increase the fruit yield, fruit quality like large, round, uniform size, deep red color and increased shelf life. Breeding for disease resistance like fusarium wilt, lead blight, anthracnose, bacterial wilt. Breeding for insect resistance, fruit borer and whitefly. Breeding for abiotic stresses resistant like cold tolerant, drought tolerant, salt tolerant, low temperature tolerant, herbicide tolerant. To breed varieties for prolonged storage and transportation. So, example that is Flavr Savr. Then last, to breed varieties suitable for processing. Greenhouse tomato cultivars and disease resistance. So, indeterminate cultivars are preferred for greenhouse production because they grow continuously, no fixed terminal bud. Produce fruits over a longer period and are suitable for staking or training system. It gives higher yield under protected condition. That's why indeterminate cultivars are preferred. Major disease resistance required in tomato. A good greenhouse cultivar should possess disease resistance to following, that is fusarium wilt caused by Fusarium oxysporum species lycopersicum. Then verticillium wilt caused by Verticillium alboatrum. Lead blight Phytophthora infestans. Early blight Alternaria solani. Septoria leaf spot, that is Septoria lycopersicum. Anthracnose Coletotrichum fomoides. Next, bacterial wilt, Pseudomonas solanacearum. Bacterial canker, Corynebacterium michigans. Then, tomato mosaic virus, TMV. then root knot nematode, Melangyne incognita. So these are some major disease resistance required in tomato. Resistant to insect like fruit borer Heliothis armigera, whitefly Bemisia tabaci. Resistant to abiotic stresses like cold set varieties, hard set, drought tolerance, salt tolerance, low temperature germination and growth, then chilling injury tolerance, then herbicide tolerance. So these are some abiotic stresses. Okay. Next, flower biology. The flowers are bisexual, radially symmetrical, and consist of 5 parts. Simple petal anthers, the calyx is united at least at the base. The corolla is also united, but its shape varies. So here you can see the flower structure. Like this is peduncle, then these are the sepals, then these ovules and ovary. Here these are the petals, and this long, these are the stamen, this is stigma, and this is style. So now from these flowers come to fruit. So seeds, this style stigma will develop into this fruit. Peduncle and sepals will be as it is. So this is the structure of flower in case of tomato. So here you can see the all the parts easily. This is the symmetrical transverse section of tomato fruit. Flowering behavior. So anthesis starts at 6 am and maximum flowering, flower open till late morning. Dehiscence 8 to 11 am. Receptivity of stigma 16 hours before and 5 days after the anthesis. Flowers are borne in small fork raceme sign. There are 4 to 8 flowers in each compound inflorescence. Flowers are pendant, perfect, hypogynous. There is a light protective anther cone surrounding the stigma leading to self-pollination. So dehiscence of anther is longitudinal, 1 to 2 days after opening of corolla. If the pollen is shed as the style grows up through anther tube, self-fertilization occurs, and when the stigma protrudes, chances of cross-pollination through bees will increase. The optimum temperature for pollination is around 21 degrees. Emasculation is usually done 1 day prior to anthesis or flower opening. At this stage, the sepals have started to separate and the anthers and corolla are beginning to change from light to dark yellow. The stigma is fully receptive at this stage, allowing for pollination even immediately after emasculation. Protection of pollinated flowers by wrapping with cotton or small pollination bags is essential. Now, breeding methods. First, introduction. So, it consists of taking a genotype or a group of genotype of plants into completely new environment where they have not been grown before. In case, for example, Cyax from USA and Marglove from USA. Next, pure line selection. A pure line is the progeny of single homozygous plant of self-pollinated species. So therefore, all the plants in a pure line have the same genotype. For example, Arca vicas, Arca savra, Arca aba, Arca alok, Arca ahuti. Single seed descent method suited for characteristics with low heritability for yield and yield components and earliness and in crops with wider spacing. So it is this SSD is the modification of bulk method. This is a classic procedure of having a single seed from each plant, bulking the individual seed and planting out the next generation. With this procedure, 1 or 2 seeds are collected from each F2 plant and then bulked to grow F3. It is a system to rapidly develop pure lines followed by selection among those pure lines. Single plant selection. So, single SPS is used to select the chance variants of types superior types from mixed homozygous population in a self-pollinated vegetable crop. This is one of the effective methods to make maximum use of germplasm. Example, Greater Baltimore and Bonnie-Best. Mass selection. The massing of IIHR 674 from VC82 line from USA developed into Arca Ashish. Pedigree method. Pedigree may be defined as the description of ancestors of an individual, and it generally goes back to some distant ancestors in the past. Pedigree would describe the parents, grandparents, great-grandparents, and so on. It is useful by finding out if two individuals are related by descent, have a common parent in their ancestors, and they have some genes in common, like Arca megali of tomato, developed from Arca vicas and IIHR 554. Pusa ruby selection from improved Meruti cross Siox. Backcross method, a cross between hybrid and one of its parents. The hybrid and the progenies in the subsequent generations are repeatedly backcrossed to one of the parent, followed when the character is governed by one or few genes. Used for improving traits like early blight, nematode and bacterial wilt resistance. Heterosis breeding. Though tomato is self-pollinated crop, it has tremendous potential for heterosis breeding. The commercial exploitation of hybrid vigor in tomato has received greater importance due to several advantages of hybrids over pure line varieties, especially in terms of marketable fruit yield, component rate, and resistance to biotic and abiotic stresses. This is why a large number of commercial hybrids have been developed. With the increasing popularity of F1 hybrids in tomato, it is essential to develop hybrids with excellent quality, high yield, and resistant to diseases and pests. Some examples of F1 hybrid like Arca Vishal, IHR 837 crossed with 932, Arca Vardhan, Arca Sreshtha, and Arca Abhijit. Interspecific hybridization. Like first, lycopersicum peruvianum, it acts as a source of resistance to verticillium wilt, TLCV, nematodes, and salinity. Next species, lycopersicum pimpinellifolium, that is source of resistance to bacterial wilt, fusarium wilt, also a source of carotene and vitamin C content. Lycopersicum hirsutum, source of resistance to fusarium wilt, helicoverpa, insect and viruses. Lycopersicum cheesemani, good source for salinity resistance. Then Lycopersicum penile, source of drought resistance and salinity resistance. Mutation breeding, that is S2 Pusa S12 Siox variety is exposed to mutation. Pusa Lal Meruti from improved Meruti. Seeds were exposed to 15 to 30 kV of gamma rays. Natural mutants are also available in tomato like NR never ripening, RIN single recessive gene ripening inhibitor, NOR non-ripening, ALC slow ripening. So these are the natural mutants available in tomato. Polyploidy breeding. Not much scope of polyploidy is there in tomato. If needed, 0.2% colchicine is used. Tetraploids will come sterile and fruit size will also come small. Biotechnology. Antisense RNA for polygalactoronase PGU enzyme, where a cell wall degrading enzyme activity appears at the onset of ripening, and in ripe fruit it is one of the major cell-bound protein. So with this, one variety is available, that is Flavr Savr. Second, viral coat protein. Introduce gene which can synthesize the protein that coats the virus material. Varieties Avinash 1 and Avinash 2. Then herbicide resistance. Gene which can resist that herbicide, means variety should not be affected but weed should be clear. Example, BT for a Helicoverpa, that is fruit borer resistance. Breeding for storage life and processing type. Fruits with high acidity, uniform intense red color, high TSS, resistance to cracking, elongated fruits with high yield. Plants should be adapted to mechanical harvesting with determinate habit. Concentrate on fruit set for one-time harvesting and jointless pedicel on fruit etc. Varieties: Roma, Punjab Chwara, Arka Ahuti, Arka Ashish. Juice for juice and canning purpose: Roma, San Marzano, Red Top, HS110. Puree preparation: Endo Process 2, Endo Process 3, Rupali. Paste and ketchup: HS101 and HS110. So this biotechnological approaches already we have seen. Genetically modified tomato, Flavr Savr. So Flavr Savr is genetically modified tomato. It was the first commercial grown genetic engineered food approved for human consumption. A genetically modified transgenic tomato is one whose genes are altered using genetic engineering techniques. The first commercially available GM food was a tomato engineered for longer shelf life. The modifications target the PGU enzyme, reducing the fruit softening and ultimately delaying ripening. Some key points. Flavr Savr was first developed by Californian company Calgen. Characteristics: developed using genetic engineering to slow down the ripening process, thereby reducing the softening while maintaining the natural color and flavor. Enable tomatoes to ripen on the vine and still tolerate long distance transport without becoming too soft. Achieved by introducing the antisense gene that suppresses the PG enzyme. Normally, this enzyme degrades pectin in cell walls, leading to fruit softening and increase the susceptibility to damage and fungal infection. It improves shelf life. Hence, fruits still require normal harvesting practices similar to conventional tomato. Breeding for disease resistance, like bacterial wilt, Arca allo PI 127805 that is Lycopersicum pimpinellifolium. For leaf curl virus, Lycopersicum peruvianum and Lycopersicum hirsutum. Resistant varieties: Nandi, Vaibhav, Sankranti. Nematode: MI gene carries resistance to nematodes located on chromosome 6 in tomato. This PI 128657, Lycopersicum peruvianum and Nematex not infected by three races of archaea. Arcavardhan F1 hybrid, then NTDR1, PNR7 and PUSA 120. So hybrids: determinate type ARTH3, PUSA hybrid 2, NARF 101, Swarna 12, NDTH6, Nath Amrut 501 and DTH4. Indeterminate type ARTH4, FM2, KT4, MTH6, FM1, Nath Amrut 601, NDTH2. Other hybrids like Pusa Divya, indeterminate type. Pusa Sadabahar, it can set fruit at the wide range of temperature, that is 6 to 30 degree. Kashi Hybrid 1 and Kashi Vishesh H86 from IIBR Varanasi. Pusa Hybrid 8, determinate hybrid from IRI New Delhi. Arka Ananya, semi-determinate and resistance to leaf curl virus. For tomato, we need warm season climate as tomato is warm season crop. Optimum temperature for cultivation is 20 to 24 degrees centigrade. Optimum temperature for fruit setting 15 to 20 degree. Temperatures below 16 and above 27 are not desirable. Ideal temperature for development of red and yellow color 18 to 25. Lycopene responsible for red color is highest at 21 to 24 degrees centigrade. Its production declines rapidly above 27. As far as the seed rate is concerned, for nursery raising, open pollinated variety with 300 to 400 gram per hectare and for hybrids 125 to 175. Time of planting: tomato is day neutral plant, can be grown in any season. Up to 3 crops per year in the frost free areas. Kharif in July transplanting and Rabi transplanting October to November. So seed treatment: Treat seeds with fungicides such as Captain, Ceres and Thyram at 2 gram per kg. So as we have seen, rainy June, July sowing, winter September, October and summer January, February. In hills, sowing time depends on elevation. Lower hills, February, March. Higher hills, March, April. So this seasonal scheduling ensures optimal temperature conditions for germination, flowering and fruit set. Irrigation. So first irrigation is applied as soon as the transplant complete. Subsequent irrigations are made at 8 to 10 days interval as per the requirement of the crop. In winter, plants are not irrigated during ripening of fruit to retard the process, but they are irrigated during summer to promote the process of ripening. In autumn, field moisture plays a vital role in incidence of leaf curl virus diseases. Methods of irrigation commonly practiced in tomato, that is furrow method, drip method and sprinkler method of irrigation. Interculture operations and weed control. For raising a successful crop, gap filling, hoeing, earthing up, staking, pruning and mulching are required. So practices like hoeing, earthing up, mulching, pruning, caging, trellis and staking. So these are the intercultural operations. Weed control. Critical period for checking weeds is 35 to 40 days after transplanting. Timely hoeing and weeding is necessary. Cost of cultivation of crop may be reduced to some extent by applying the herbicides in place of manual weeding. Pre-transplant surface application of Alaclo 1 to 1.25 kg per hectare or oxyfluorphan 1 kg per hectare or flucloraline 1.25 kg per hectare is recommended. Post-emergence application of metribuzin 0.5 kg per hectare may be effectively applied. Yield. So yield of tomato depends on many factors like soil, variety, season and management practices followed. However, an average open pollinated variety yield 300 to 500 quintal per hectare and hybrids 500 to 800 quintal per hectare. Future prospects like conventional cultural practices and mere application of chemical fertilizers are not enough to fill up the gap between the production and demand. Therefore, sustainable and low-cost technologies for tomato cultivation should be developed, and several conventional techniques of tomato production must be modified to suit existing local conditions based on practical experiences and status of resource-limited farmers. Moreover, future tomato breeding research should be continue to be driven by some of the same factors that drive it now. But as the global weather pattern changes, their impact upon growing conditions should require greater adaptability in varieties. Attempts should also be made to undertake in vitro culture, molecular biology, and genetic transformation. Last part, that is conclusion. From first domestication to modern breeding, the tomato has been continually subjected to human selection for a wide array of applications in both science and commerce. Current efforts in tomato production and breeding are focused on discovering and exploiting genes for the most important traits in tomato germplasm. Besides trying to develop resistance to familiar diseases, researchers are increasingly focused on new pathogens like Gemini virus, a disease now limited to tropical regions, and if spread to other growing regions could be devastating. In future, new approaches, ideas and technologies will emerge which will affect the direction of production. So, this is all about the tomato breeding and tomato seed production. Some basic information for tomato seed production. So, here we have seen like what are the breeding methods in tomato breeding, what is the flower structure, right? Flowering behavior. Then what are the objectives of tomato breeding like earliness, fruit yield, fruit size, fruit shape. Okay. Then what are the different major disease resistances required in tomato? Okay, then resistant to insect like major insects are fruit borer and whitefly, and resistant to abiotic stresses like cold set or hot, then drought, salt, then different breeding methods, then breeding for storage life and processing type. This is the most important when in case of transportation, the shelf life. So that's why you need to work on the shelf life. Then some biotechnological methods like antisense RNA technology, then viral coat protein, herbicide resistance through biotechnological approaches, and insect resistance. So in insect resistance, example our BT cotton, which is resistant against Helicoverpa armigera, pink bollworm. Then we have seen the Flavr Savr tomato. Okay. Then there's some resistant sources to different diseases, characters of some hybrids developed by public sector. Then we have seen climate, seed rate requirement, then season for tomato cultivation, how to give irrigation, with some agricultural operations, weed control, and at last we have seen the yield. So this is all about tomato. Hope you will get some valuable information from this. So till next session, it's thank you very much. Thank you.